come later, but I'm not sure. Usually we get more people here, but thanks for coming out tonight.
Of course. Thank you for having me.
So let's start out with some questions. Who has questions?
I just have a general question. If the slides, like, from our notes are not on this one, will they be on the test?
No.
Okay.
Yeah. So again, the whole idea is that it should be.
Okay.
Yeah. Yes.
I have a question that, like, you said, like, some questions are long. So, like, how long we have to explain, like, the question if it's long?
I try to write the questions so that they're not real long, or they're going to be worth more points, right? So if it's, I can't remember how many points there are. 70 points. If something's worth 5 points, it's going to be longer. But the idea is, typically, if you sit down and think about, and you know what's going on, and you think about your answer, you should be able to do it in a few sentences, usually, right? So most people don't have any problems finishing. Yeah. Okay, so let's. I'm just going to go through the slides. Hopefully, maybe. I'm going to turn this a little bit so maybe people can see where we are on the slides as well. Okay, and we'll just, we'll come to stuff. If you see something, you want to ask a question, do so, right? And there's a few things I'll probably focus on that are, I know are harder. Some of them are just easy, right? You should be able to do this. Nothing too crazy there. Should be able to do all the different parts of the layers of the skin. Remember we talked about these are different general regions, and then we can go into the epidermis. We talked a lot about the layers. Right? And so you should be able to know the layers of the skin. Right? A lot of this you're going to have to know for the practical too, so there's nothing too crazy there. Right? Hopefully. Yes. Yeah.
Will we have to be, like, drawing pictures up for the exam?
Not necessarily. There's some things I ask you to draw a picture, but also we'll see if you want to just do a description instead.
Yeah, okay.
Yeah. Besides, some people aren't great at drawing.
Yeah.
Other people are. So, I realize there's differences. All right. So the keratinocytes make what? What's the thing that they make?
Keratin.
Keratin, which is what type of structure? It's a protein. What's it like? Why do we have keratin?
For, like, our hair and nails.
Yeah, but why? What is it that makes it so sort of important for hair and nails? So remember, it's very structural. It's a really rigid protein. It's really hard to degrade in any way. So this is why like hair, your drains get clogged by hair, right? It's really hard to get rid of it. Very tough protein. Stem cells. Right, we talked about two different niches. Right, niches are places where we find stem cells. One of them would be the strontum basal. Where else do we find stem cells? We actually talked about more than one. Hair bulge, right? It's another place. We talked about the osteogenic layer of the periosteum. We would also find some stem cells there. There are stem cells in the red marrow that are going to help make osteoclasts. Right, so forth. All right. What are Langerhans cells? A little bit different, right? What do they do? Okay, so they're immune cells. So again, these are. I think it's some sort of open mic or something.
Hello.
Is that a sign from above? All right. This review is a little bit hard to do because it's all very factual, right? So let's see if we can find something that's not just facts. No, that's pretty much just facts. Okay. So the idea of skin tone. So some of the classes this came off better than others. Why do we have various levels of melanin? What are we trying to balance? Well, if we think about it, let's group this board up. We have these cells that need to have melanin in front of them to protect their DNA from damage. So if you live closer to the equator, or if you say your ancestors live closer to the equator, you're typically going to have darker skin because you were exposed to more UV radiation. right? And having, of course, exposure to UV radiation would lead to more skin cancer, so it's protective to have this melanin. Now, we want melanin to be protective, but the other thing we want to do is that we need a little bit of UV radiation to do what? We need a little bit of UV radiation to pass through here to help us do what? And it's in the blood. Why do we need some UV radiation to make its way through? Vitamin D. Okay, so we also said that you have a molecule that's a precursor to vitamin D that's floating around in the blood, right? So this would be in the dermis. And we need some UV radiation to hit that and help us make vitamin D. And again, as you'll see from the next slide, that's a pretty complicated process. What I want you guys to know is that it really is, we activate this precursor molecule with UV radiation. So if you have very dark skin and you go to Alaska, well then you're blocking too much of the sun. Right? You're not having anything get down here to help us make vitamin D. Right? If you have very pale skin and you move to Australia, well, you're not getting enough protection. So there is this little balance between protection and our need to make vitamin D. Key thing a lot of people confuse, we don't get vitamin D directly from the sunlight. So people sort of like, we're getting bombarded by vitamin D molecules. It's the fact that the UV radiation helps us make vitamin D, right? And why do we care about it? What does vitamin D help us do? We talk about this condition, rickets, and bone softening, right? So vitamin D helps us deal with calcium. So it helps with production of healthy bone. All right. Lines of cleavage. Again, this idea that if you cut across the lines of cleavage, that's going to lead to more scarring than if you cut with the lines of cleavage. The lines of cleavage are found in the deep layers of the dermis, so down into the reticular layer. As we go deeper, we see larger and larger cables of collagen, and those are the lines of cleavage. Should be able to draw these two different types of glands, so this is something I think everybody can draw. Right, so could you draw these glands? Okay, so, tube, right, and the other one, like a lobe, very, okay? So I think even Emma might be able to, can you do it?
Wrong?
Yeah,
Yeah.
okay. I know, maybe you're really good at it. Are you really good or no?
I had to for Dr. Zimmerman last semester.
Yeah.
Drew a lot of pictures for those exams.
Did they come off well?
Yeah, I'd say so.
Okay. Okay. The divisions of the sympathetic and parasympathetic division of the autonomic nervous system. Right, you should know those two things. And we had the fight-flight response and the rest and digest. Most of the stuff we talked about was under the control of the sympathetic, right? We talked about the erector pili. I think that's really the only thing that we talked about. Oh, and sweating is also under the control of the sympathetic. Okay, so let's step back a little bit. So we talked about, besides the ANS, we talked about some other general control systems. So one of them we talked about were sex hormones. We talked about androgens. And estrogens. So androgens are related to what? What did we talk about? So we actually kind of these are each involved in different systems. So the androgens we talked about in context of what? Male sex hormones, but what did they do? What the things that we talked about, what do they control? Well, a little bit. Yeah, so the androgens are mostly involved in what we discussed in the integument, right? And what we discussed in class, again, there's more to it than this, but what we discussed in class is the estrogens are involved in the skeletal system. So give me an example of something that happens with androgens in the integument.
The hair turning terminal.
Terminal, right? Terminal hair development. You guys just went through puberty not that long ago, right? so what? You guys don't remember any of this? What else? Why do you have to worry about body odor?
Sweating.
Sweat, not so much, well, part of sweating. What part of sweating? Pheromones, but, well, yes, probably, yeah. Well, they're made by what gland? The apical gland, okay, which is special sweat glands that we think, might be making these pheromones. Then, what else happens? Why do you get, like, whiteheads and blackheads?
Well, it like builds up in your.
So what glands are doing that?
Spacious.
Sebaceous gland, right? So, well, they're always there, but you make more sebum, right? So those are, again, androgens. Estrogens, we talked about this mostly in the context of the skeletal system. One of the things we discussed is that it's protective in the sense that it helps osteoblasts remain active. And it keeps down the number of osteoclasts. So there's less of them around. What else did estrogens do for the skeletal system? Yeah. What specifically, we talked a little bit about there's differences how different bones react to androgens and estrogens, but we talked a lot about the context of long bones. What does estrogen do for long bones? Okay, closes the plate. What else?
Prevents osteoporosis.
Yeah, but we're talking about development now. So it not only closes the plate, we talked a little bit about why girls have a growth spurt before boys. We also, so in other words, if estrogen levels get to a certain level, that's going to trigger a growth spurt. It affects the epiphyseal plate, causes it to grow more. Once those estrogen levels get too high, then the growth plate stops growing. So it does sort of both, right? Closes plate and it sort of activates the plate at puberty. So really, it's just kind of how much you know. Okay, we talked about sweat and cooling. Those are friendly apical glands. Sebaceous glands. Vellus and terminal hair, we've already covered that. Okay. All right. What's the purpose of the hair bulb? Or, sorry, hair bulge, right? What's the purpose of the hair bulge? Okay, so again, just to make it clear, the hair, they're stem cells. So the hair bulb is the whole general bottom of the hair. The hair matrix is where the cells start to grow. So it's basically the equivalent of the stratum basal. But there is not a consistent number of stem cells there. Those stem cells eventually die. So they're not really stem cells in the fact that they die. And so that the population of stem cells that are around all the time are live in the hair bulge. And we have to get new stem cells every once in a while to repopulate the hair matrix. All right, so you guys pretty good with different cancers, tumor, benign, malignant, metastasis, right? You should know the 3 different types of skin cancer: basal cell, squamous cell, and melanoma. The most serious is what? Okay. And what cells does melanoma come from? Melanocytes, right? And there's a lot of melanocytes, in a freckle or a mole, so there's a higher chance of developing something there. You should know the ABCDs and E's, melanoma. First, second, third degree burns, right? First degree is only the epidermis is affected. Second degree, the epidermis is gone, but the dermis is affected. And then a third degree, both epidermis and dermis are gone. Right? So we talked about third degree being critical, and I think we did the rules of nines. You should be able to do that. But when we talk about dehydration being an issue, what's the problem with dehydration? It's not just a lack of water. What's the major problem? Yes, to change electrolytes. So we lose electrolytes, and that actually changes the function of muscle first, and then eventually cardiac muscle, and it can lead to heart arrhythmias, and finally heart failure. Right. Rule of nines. Again, I think you guys could do this. This might be something that could have you draw. I don't know. Okay. Bones, cartilage, joints, tendons, ligaments. You should be able to do this, right? We did this kind of in lab as well, right? So let's talk a little bit about long bone development. This is one of those things that gets a little confusing. And I think I actually had people draw this at one time. I think got really crazy. So I'm going to have to figure out a way to ask this question without having people draw it. Huh?
They got crazy.
Well, I mean, you try to draw like, just little teeny bones and little cells everywhere. Okay, so let's start off. So again, we'll go through several different processes here. Again, it's best to kind of think about things conceptually. And so for most of the bones that we talked about, we start out with cartilage. And then we're going to have some sort of calcification, which really just means depositing crystals onto collagen. And then we're going to have some sort of conversion to bone, and then often some sort of remodeling that happens. Right? Those are the 3 C's that we discussed. So we're going to start out with the bone formation. So for the test, I don't expect you guys to know exactly when bone formation starts, and we did go through several weeks, like here's something that's 12 weeks, just to give you guys an idea of what's happening. The only timing that I want you to know is that the development of the epiphyses occurs right before birth. It's a fairly late process. So if you know that's in the last month or so of development, that's fine. It kind of gives you an idea of what's going on. So again, we're talking about long bones, and everything starts out as a little bone made of what? Why am I drawing that in blue? Other than the fact that my black marker's not great. Yeah. Hyaline cartilage, right? So it's a little miniature bone made of hyaline cartilage. Again, it really applies to the long bones. If we're talking about the skull, that's an example of a membrane bone. I think that made the cut, right? So you should know that a membrane bone is one of these flat bones that does not develop from hyaline cartilage. So what happens next? Well, that black marker is just horrible, all right? I'll use green for bone. We developed this little thing that wraps around the bone called bone collar, okay? So in order for that to happen, that has to develop some from osteoblasts coming into the area, right? And what we're going to do is we're going to start developing this bone collar. Now the bone collar is cutting off this central area from nutrients. So the next thing that's going to happen is that we are going to have some crystals form. Right? That's the calcification we talked about. And again, it's happening because the cells there are having a hard time maintaining collagen. It's an active process to remove calcium from collagen. And as they start to lack energy, because it's harder for them to get nutrients because of this collar, they can't really maintain the collagen, so crystals develop, and we get a little cavity form. Right? The crystals kind of fall apart, we have a cavity form. The next thing we want to do is to bring in some blood, right? Because if we want to convert this area to bone, we have to bring in osteoblasts and osteoclasts. So we're going to bring some blood in. What's that little blood vessel called? Well, if we talk about a bone, it's called the nutrient foramen. Developmentally, this is called the periosteal bud, okay? So that is going to bring in osteoblasts and osteoclasts, and we're going to start making bone. Now, what kind of bone do we make? Spongy bone, right? Because we don't really have the time if we developed compact bone, that would take a long time, and we would leave the bone in a fragile state. So we're going to develop some spongy bone in the center, okay? Which means that eventually we have to destroy that spongy bone, or I should say rework it slowly, right, to make a compact bone with a cavity in the center. Now, everything we've talked about so far is starting on the diaphysis, or shaft of the long bone. Because this is the first place that this bone is starting to form, we call that the primary ossification center. And what's going to happen over time is this guy's going to get bigger, right? The bone collar keeps on getting bigger. We just have a recapitulation or a redo of what we've done so far. We keep on having calcification. We keep on making a cavity. We have spongy bone here, right? Where things are just getting longer. And then about a month before birth, we're going to have the formation of our secondary ossification centers. And this is going to be where we're going to start having just the area kind of basically get calcified. There is no cavity that forms, but we bring in osteoblasts and osteoclasts, and we make spongy bone. So that's our secondary ossification center. And again, it's really about 1 month before birth. So we bring in osteoblasts, we bring in osteoclasts. What else has to come in here? When we think about it, that spongy bone is going to be filled with what type of cell eventually? What's the major function of the epiphyseal spongy bone, the area between the spongy bone? Red marrow. So and what do we do with the red marrow? We make blood cells, right? So we have to bring in stem cells into this area as well. So the stem cells are going to help us make blood, are also going to arrive. So if you could do those steps, and again, I don't expect you to draw them, then you're in pretty good shape. Right? Those are the major key things that happen. Okay, so let's look at this area, because if we look at a bone that's a little bit older, we would see that we have an epiphyseal plate that's left over, right? So let's talk about this guy. So let me go a little forward in the notes, because it's been a while, I think it's been like 4 years since we ever covered this. Yeah, because we usually have to cut stuff because we're behind schedule. Right? You guys should be able to do this. Huh?
Yes.
Yeah, well, it's not you, it's the weather, I think. Yeah, so it's a little bit unusual. Okay, so this is what I'm talking about. Okay. All right, so we have our epiphyseal plate. Right, basically made of hyaline cartilage. And we have arresting stuff going on. Right, we have an area where we have a medullary cavity down here. There's blood vessels. Okay, both these areas are sending signals that are going to drive this process, because you're going to see that, as we go through different things, the cells are going to change their form. It's not strictly like it was in the skin where things change their form because they're moving away from nutrients. It's that they're receiving signals from these two sides, and as they move, the mix of signals they receive changes. Right? So, for example, if we're here, we're receiving a lot of signals from the resting zone, not much from the blood. If we're here, we're receiving a lot of stuff from the blood, not much from the resting zone, and everything in between. So that's what's driving the changes in the cells that we see. So the very top, we have some rapidly dividing chondroblasts. In what is called the proliferation zone. How do I know they're rapidly dividing? What's a physical thing that you can see in this picture that tells you they're dividing rapidly? right? They're up here, incredibly small. So they're not dividing and growing. They're just dividing, and they get to a certain size and they split again. So they're really not taking the time to grow. That's why these are so small. Then they start to grow. Right? That is the hypertrophic zone. And they get so big that sometimes the little areas start to fuse together, right? Remember, these are chondroblasts, but there's a little lacuna around them. Sometimes the lacunas can combine. Right? In the next area, you'll notice that we have some calcification occurring. You can see these little crystals forming, right? And as we said, what's happening is that they are sort of dying here, but it's not because of lack of blood. It's just the signals they're getting. They're not taking care of collagen, and so you start to have these crystals form in this area. This is the calcification zone, which kind of goes with our 3 C's that we talked about. At the very bottom of this, right, the crystals kind of have a stalactite appearance that are called spicules. And really into that area, we do have a blood supply that is carrying osteoblasts and osteoclasts. Right? And they're going to convert these spicules into bone. And so the area where we have this conversion of spicules into bone is where we call that our ossification zone. Right? Or osteogenic zone. So what kind of cells do we need to have that occur? So here's your spicules. What kind of cells do we need to make bone? Osteoblasts and osteoclasts. Always those 2 things together. What kind of bone are we going to make? Spongy bone, right? Again, if you're thinking about what kind of bone we make, remember that spongy bone takes about 1/10 of the time to produce that compact bone takes. So we want to stabilize this area. We're not going to take the time to make compact bone. We're going to make spongy bone. Again, if we come back 3 years later, this bone's a lot longer, we might have converted that to compact bone, but we're going to make spongy bone first, okay? Okay, so that's another type of thing that's happening. Let's do the final thing, which is repairing a fracture in the long bone.
I have a question.
Yeah.
In the picture that we drew in class, you added these arrows that were like A signal and like B signal.
Yeah.
What are those?
Well, that's where I was talking about that you have signals coming down from the resting zone.
Oh, okay.
And you have signals coming out from the blood supply in the bottom, right? And so, so it was kind of getting at. So if you had A signals and B signals, this area was getting a lot of A and a little bit of B. This area is getting a lot of B, a little bit of A. This one's getting about, the same of both.
Okay.
So there's a mixture. So this is what we call a gradient, or a developmental gradient. That's something that happens a lot in biological systems. All right, so we have a fracture. We break a lot of blood vessels. So what's the first thing that happens? We get this nasty blood clot. What's that called?
No.
No, you're close. It's a swelling. Hematomas, right? A blood swelling. Oh, is this, you guys have midterm?
Yeah. Mm-hmm. Yes.
It's only every other week, isn't it?
No. It's switched it. Yeah.
Oh, they changed it?
Mm-hmm.
It used to be like once a week for 2 hours, every other week for 2 hours, so.
Yeah.
Okay, well, that's good. All right. You have a hematoma. All right. So what do we have to do first? Right, we have to reestablish a blood supply. And I'm going to have to go back to blue. Right? So we reestablish a blood supply. We talked about bringing some different cells in. What kind of cells did we talk about bringing in? Osteoclasts and blasts. We talked about making phagocytes to help us destroy tissue. Now, one thing we have to think about, are we actually ready to use osteoclasts and osteoblasts yet? What do we have to do first? So go back to our 3 C's. What do we have to do first? We have to make cartilage, right? So we do have some chondroblasts that come in as well, right? And they're going to be the ones that are active first. So what type of cartilage do we make? Hyaline cartilage is not that tough. Fibrocartilage, okay? So we made fibrocartilage, and we said that this fibrocartilage, in the context of this bone, has a certain name to it. What do we call that? Fibrocartilage connection. Something with callus. All right, we're going to make a soft callus. Right, and again, this is coming in through a reestablished blood supply. We make a soft callus of fibrocartilage. Doesn't take very long for us to make that. Right, really about a week and a half, depending on how bad the injury is. It's not something you can move around, like you can't put your weight on it, but it's not like your leg's going to be swinging around like that, right? So what we're going to do over time is we're going to have the fibrocartilage develop crystals and use the osteoblasts and osteoclasts to convert those crystals to bone. So that's our calcification followed by conversion. And what type of bone do we make? Soft, I should say spongy bone. And that spongy bone has a name. What do we call it? What do we call that? Think of spongy bone that's in the gap. Yes.
Oh, I didn't know.
Oh, I thought you were saying. I thought you had your hands. Okay. It's another callus. This is what? Hard callus. Okay. And then over time, that's reworked into, in this case, there'd be a medullary cavity there with compact bone around the center. Now that compact bone is going to be produced in response to the stresses, because if this was a normal fracture, at about 2 months you'd start putting your weight on this. The area would start encountering mechanical stress, and that mechanical stress would be sensed by the osteocytes. They send out signals to recruit bone remodeling unit, and we would reform this to deal with the stress. That process, where we have bone become stronger when it's exposed to stress, is called what? That's Wolff's Law. Right? And we gave a lot of different examples of that. You guys should be able to do, at least 1 or 2. Okay. Here's an example of gigantism where we have excessive growth hormone before the epiphyseal plates close. Right? What happens if you start taking growth hormone after the epiphyseal plates close? So let's say I'm 23 and I decide I'm going to start taking some growth hormone. What happens? There's a condition where we see excessive growth hormone. Remember talking about Andre the Giant? What was that called? Acromegaly, right? It gives you sort of a brutish, rough appearance. It's because the bones in your skull are starting to grow. The bones in the hands and feet continue to grow. We discussed the fact that it also causes some of your internal organs to continue to grow, including your heart, and that's not good, right? So people with acromegaly, if they're not treated, typically die in their mid-50s, if not before that.
And that's after the plates are closed.
After the plates are closed, yeah. So you have some people that are a little bit like a mixture, right? So Andre the Giant, for example, had some gigantism, and I don't know if he's still growing when he died, but he was definitely had progressive acromegaly, right? If you look at pictures of him, he clearly was changing over time. Some of his bones were still growing. All right, what happens if we don't have enough growth hormone? Then we talk about you get chihuahuas. We talked about dwarfism. And so again, we just said the most common type of dwarfism is achondroplasia, where the chondroblasts that are making, that are making the new cells in growth plate are not detecting the proper signals to continue to grow, so you don't have growth in long bones. Different process in the flat bones, the skull, for example, so they continue to grow, just the long bones that don't grow. So we did Wolff's Law. homeostasis. So when we talk about stuff in class, if depending on what class you go to, you'll see that I'll write something on the board, and often I'll write a slightly different definition, because the important thing is that you understand the concept. So the concept for homeostasis is that there's something we're trying to maintain within some sort of narrow range. So in the particular case we're talking about now, we're going to talk about PTH and the fact we're trying to maintain calcium levels. So we're trying to maintain something. And the second part of that is that it's an active process. We wouldn't really have to have homeostasis if it was something that never changed. So this is a value that's always going to change. We don't want it to go too high or too low, so we have to have multiple processes to make sure that doesn't happen. Right, so the one we talked about is the fact that if calcium levels fall too low, if you have hypocalcemia, you're going to trigger the release of parathyroid hormone, or PTH. And this, of course, is from the parathyroid glands. Right, and again, what's going to happen is the PTH levels are high. What happens if PTH levels go up? Well, the osteoblasts sense that, right? They have receptors for PTH, and they're going to activate the osteoclasts. The osteoclasts are going to go destroy some bones, and calcium is going to be liberated, so blood calcium levels would go up, and then the PTH levels would go down because these little organs are sensing the fact that calcium levels have rise, so they don't need to release as much PTH. Right? If you have high levels of calcium, there's another mechanism at work, but we didn't really discuss that. It's not as well understood. All right, where else would we find PTH receptors? How else could you increase the amount of calcium in the body? Through diet, right? So we talked about there's some cells that would detect PTH, and those cells would be in the small intestine, and they'd enable you to more efficiently pull calcium out of your diet. You guys are constantly peeing out calcium, right? So if you could make it so that you don't pee out as much calcium, calcium levels go up. So in the kidney, there are some cells that would pull calcium back into the blood so it doesn't go to the urine. So they also have PTH receptors. The key thing here is that there's almost really very few places that have PTH receptors. So very few cells are affected by PTH. In contrast, we talked about some hormones like growth hormone and thyroid hormone that are broadly effective, right? Growth hormone did what? So, think about Barry Bonds using growth hormone, allegedly. It wasn't so he could get a bigger head. Why was he using growth hormone? For muscle, right? So a growth hormone makes skeletal muscle and the skeletal system produce more protein. Those cells are going to produce more protein. A few other connective tissue areas are going to produce more protein. And so, that's more structural elements. Yeah?
For the PTH stuff, do we need to know about OPG and rank L?
I think it's a slide, isn't it? I think we'll get there. Yeah, I think it is a slide here. And then thyroid hormone is involved in
Digestive?
Very what?
Digestive system?
No, not necessarily.
So endocrine.
It's endocrine, but it's basically going to affect the citric, I don't know if you guys call it the Krebs cycle or citric acid cycle or what you guys call
Krebs.
it? Krebs. So it's basically going to make all the enzymes in the Krebs cycle more efficient. And so every single cell is going to make more ATP, and you can use that energy to do things. Right? So it's a very powerful hormone. And basically every cell in the body is, with like one or two exceptions, is going to be able to detect thyroid hormone and respond. Okay, so we did that. Okay, OPG and rank L. Right, so what do they do? So first of all, which cells actually that we discussed is making OPG and rank L? Osteoblasts, right? So it's really the osteoblasts that are doing this. And what they're going to do is they're releasing these signals that are either going to promote the activity of osteoclasts or inhibit the activity of osteoclasts. So I'm going to use green for go, right, promotion, and red for stop. So which one of these, rank L or OPG, tends to promote the activity of osteoclasts? Rank L, right? So if there's high levels of rank L, right, and this is one of the things that goes up whenever we have PTH, this increases osteoclast production. Right, remember, if we go back to that slide I showed you, we're looking at sort of the development of osteoclasts, and basically every step is pushed towards development with rank L and with activity, right? So we have an image, we have a mature osteoclast that's just inactive. It sees some rank L, it becomes active. The OPG decreases the number of osteoclasts that are made. And decreases activity. So if there are low levels of PTH, which one of these hormones, actually no, they're not really hormones, they're just proteins, which one of these proteins should be high? Again, there's low levels of PTH, so which one of these should be high? So if there's low levels of PTH, do we want osteoclasts to be active or not? We want them to be inactive, so which one of these would we release? Yeah, we'd have high levels of OPG, low levels of rank L. Right? PTH levels are high levels of rank L, low levels of OPG. And I gave you the example of trying to drive a car with having your foot on the gas and the brake at the same time. Right? That's kind of what's going on here. But this is actually meant to work, whereas doing that with your car is not a good idea. Okay, osteoporosis, you guys are pretty good with this. Resorption, again, remember, is the process of breaking down bone and resorbing the materials. That's the osteoclasts. Deposit is making new bone. That's the osteoblasts. So as we talk about the osteoporosis, what's going to happen is that we're going to be breaking bone faster than we make it. And as we discussed, one of the reasons this is more common in women than men is that as estrogen levels fall around menopause, that means that the check or the brake that we're putting on osteoclast production is going to go away, so we're going to make more osteoclasts. And the fact that we were keeping osteoblasts active for longer periods of time, that's going to stop, and so we're going to have less osteoblasts. So we're going to be destroying more bone than we make. Right? The disease that we talked about where we have too much deposit was called what? Too much deposit of bone. What disease was that? Paget's disease, right? So we talked about two different types of Paget's. Early stage Paget and late stage Paget's. Most people are only ever going to see early stage Paget's. And what's going to occur there is that we're going to have an overactive bone remodeling unit. So both osteoblasts and osteoclasts are too active, and they're just randomly making bone. They're not making bone under the direction of Wolff's Law. Right? They're just making bone. And so what you see is a breakdown of normal bone, right, and the development of this Paget's bone, which is a very weak bone. It's kind of like spongy bone, but it's not patterned like spongy bone. It's not patterned in a way that is going to deal with forces. So this is going to lead to more fractures. So superficially, somebody that has Paget's versus osteoporosis, you know, they might present with sort of the same symptoms as far as like, well, they start having fractures. And it's only when you do the x-ray do you realize, well, this is not osteoporosis, right? This is actually Paget's. What happens in late stage Paget's?
Osteoblast more active.
Yeah, so the osteoclasts, not the osteoclasts, the osteoblasts, right? So the osteoclasts kind of go back to normal activity. the osteoblasts remain hyperactive. So we're actually going to start adding bony mass, right? So here the mass isn't really changing. We're just rearranging the bone. We're going to start remaking bone, right? And you'll start to see bone on the skull. Sometimes the ribs will start to grow extra bone. There's all this kind of stuff that happens, right? Again, the only people that have this are people that typically get patches early, or they just happen to live a really, really long time, right? It's one of the diseases that most people develops late in life. So they never typically get to this stage. Okay, so well, this seems easy.
Yeah, doesn't it?
All right, so there's a lot to learn here, right? But most of it is kind of straightforward, I think. Right? There's some processes you guys are gonna have to learn, but some of it's just like, here's a layer of skin, right? What is it? You guys have any questions about things that we didn't talk about?
Will there be any like labeling of like? You give us a picture with the label what it is and stuff.
Oh yeah.
Yeah.
I was seeing if you would
It's
tell
the
us.
yellow one with the osteo. We talked
Oh,
about glasses, like
with the
I
like
would
little
very,
spiders.
like look at this photo a lot.
Yeah.
Oh, okay.
Yeah, and there's I mean, yeah. So something like that. So sometimes I'll print it on the test. Something like that is usually I'll project it on the screen
Okay.
because you just the copies can get a little
Yeah,
tough.
that's the one.
Yeah.
I say it's
Yeah.
spiders too.
So you'll see there's several other pictures in here that are pretty easy to label, right?
Mhm.
Like that would be easy,
Yeah, just that.
or something like it, right?
Oh yeah.
Let's see, where's another one? Well, that one could be okay. It's a little bit. I usually find one that's a little bit like that one, right? Or something like it. You get the idea. So those questions are typically meant to be somewhat easy, so people get points,
Okay.
right? They're not, and they're because they're also something like you do. There's something like what you do in lab as well. So, so like I said, there will be a little bit of everything on the test. There'll be some multiple choice. There'll be some fill in the blanks. Be definitely short answer, identifying things. There'll be some that are a little longer, at seeing if you understand some stuff. There'll probably be some stuff where I want you to define something, but it's kind of like Wolff's Law or homeostasis or stem cells or something like that, right? Somewhere we talked about it, and you guys should come up with your own definition for it, right? Okay. All right, so unless you guys have any other questions, I guess we'll call it a night. Yeah.
Would you talk about lab a little bit?
Please do.
I have that tomorrow. And
I have it tomorrow too.
All right, so what about lab?
Everything. Like all of it.
Well, I mean, the thing is, think about the stuff we spent time on in lab. It's a little hard to tell you exactly what to study, but think about the different stations we did and what we did at those.
I've been reading the lab manual.
Well, that's a little helpful, but I would think about, like, there's stuff we didn't spend much time on, right?
Yeah.
So, and you got to think about, like, what could we easily test you on?
What could you?
Okay, well, think about, let's go back to the last thing we did, right?
So long bones.
Long bones, right? You could easily say you could be able to identify stuff on long bones. We could have pictures of different cartilage, right, and ask you what they are, which one is which. We have that Osteon model that some of you guys have spent time on.
Yeah.
The animal cell.
So,
No, the animal cell, no like the tendon and the articular cartilage.
Do we need to do like anything for the X-rays? Like I know the
Yes.
fractures, but do I
There's
need
gonna
Yeah,
be
sure.
like 2 or 3 questions.
what? How do you know this?
I took
It's
this.
okay.
I had to drop it. It was
Ow.
so
Oh, you can't be doing this.
We'll speak after class.
Well, okay, but I mean, but the thing is, you should be able to kind of predict that. Now, you guys haven't had the class before, so, but by the time you get done with this first practical, you should be able to predict what's on the next practical.
The cat one has a word bank.
Yes,
Are
that's
word
right.
bites on it? Or is
Oh,
that just
No, word bites are not on.
Oh,
Okay.
okay,
Yeah.
good. Because I'm not.
You only have to know this for the quizzes.
Yeah, this is for the quizzes. So, as far as the scope stuff, we will ask you to find something on the scope. but most of the stuff is going to be pictures
Well,
for
we
the
have to do,
histology.
like, histology. Yeah, histology.
Is
Yeah.
this the one where we have to draw a picture from the scope? Or just when we have to find something? And you
I don't think we ever done that on the practical.
Okay. I'd rather.
Well, okay, maybe.
Lieber decided to change the entire thing when he first came here the first year, and it was hard.
I don't think it was
It
just.
was awful.
I don't think it was just him who did that.
It was horrible.
He's not that involved in the labs. It's probably Dr. Camwell that did that. Because we did rework. Well, that was the time we started reworking. It wasn't Dr. Lieber's fault. But I think we'll leave it at that. How long? When did you?
Well,
So will it be like the basal layer? Like this is the basal layer and this is the spacious gland.
For which one?
Like identified on a scope.
That's possible. So typically, what we'll do on the scope is we'll say, find this and
Show.
show it to us.
Oh, really? That's
Yeah.
great. Oh, I can do
That's
that.
it.